Welcome. This is the first of a series of short lectures designed to be an introduction to immunology. My name is David Leitenberg. I am the course director for the medical school course in immunology. I'm also an associate professor in the Department of Microbiology, Immunology, and Tropical Medicine, and I'm the medical director of the Laboratory of Immunology, the Diagnostic Immunology Lab at Children's National Medical Center. So this first lecture is really designed to be an introduction to the immune system, why it's important in health and disease, and also an introduction to innate immunity and innate immune cells. The objectives for this first lecture are broadly to describe the role of the immune system in health and disease, to compare the innate and adaptive immune system in terms of antigen recognition and response characteristics, to identify the names and functions of cells of the immune system and classify as to whether or not they are primarily parts of the innate or adaptive immune system, and then to describe a little bit more specifically the main features of innate immune cells and recognize the roles of complement proteins in mediating immune responses. So in general, what is the immune system? And one way to think about this is just a collection of cells and immune organs that work together in a relatively complicated way to protect against foreign pathogens. The immune system has evolved a high level of complexity in part because it has a lot of challenges in dealing with pathogens that are present in our environment. These pathogens can range from relatively large multicellular worms that you can see down to tiny single-stranded RNA viruses. And the immune mechanisms that are effective in clearing or dealing with these various pathogens are different because the nature of the pathogens and the way they infect and cause disease are different. We know that the immune system is critical for protecting us against infectious disease because there are patients that have very severe primary immunodeficiency syndromes where they're essentially born without a functioning immune system. And so this is just a 5-month-old boy who presents with a history of frequent and recurrent infections that have been attributed to multiple different types of microbes. In this case, he has a history of frequent and recurrent ear infections, bad cough, diagnosed with pneumonia. You can also see in his mouth that he's got a white exudate on his oral mucosa, which is indicative of Candida. And you can't really see in this picture, but if you shined a light in his mouth, you would also notice that his tonsils were small, indicating the opposite of what you'd expect to see as somebody with an upper respiratory tract infection that should have enlarged lymphoid tissue and enlarged tonsils. In this case, in somebody that's immunodeficient, they may have very small or not visible tonsils. Identification of microorganisms that are involved in this case demonstrated the presence of multiple different classes of microorganisms, including bacteria, viruses, and again, as you can see, fungal infections. And so there are patients like this illustrate that without a functioning immune system, we are very prone to becoming infected with multiple types of pathogens, and in severe cases where we really don't have immune system functioning very much at all, this will be a lethal disorder. We also know that the immune system is important because we've developed ways of manipulating immune function, primarily by the introduction of vaccines. Vaccines are one of the best and most successful clinical approaches that are used to dealing with disease in people, and this is just one example of many where the introduction of, in this case, a diphtheria vaccine has been shown to greatly reduce the incidence of disease, and this is all due to the introduction of this vaccine that is able to activate an immune response that's protective against diphtheria. We also know that the immune system is important in the pathogenesis and regulation of really almost any clinical disease that you can think of. We've already mentioned how important it is in protecting us against infectious disease, but you'll learn in the next few lectures and also throughout your training that immunology and inflammation is critical for regulating almost any type of clinical situation, ranging from atherosclerosis and heart disease, which is associated initially with an inflammatory response, It's also important in cancer pathogenesis, both in the terms of the immunology is important in protecting us against the development of malignancies, but also chronic inflammation is also associated with the tendency to develop tumors. Immunology and inflammatory responses are critical in promoting wound healing, and a whole variety of other fairly common clinical syndromes that you'll see. Some things that are obvious, like allergy and asthma. Other things that, like you see in this picture of a child with systemic lupus erythematosus, which is kind of a classic example of an autoimmune disease. But also it's becoming more and more appreciated. Immunology and inflammation is also associated with things like obesity and Alzheimer's disease. A whole variety of other diseases that you don't necessarily associate with inflammatory or immunologic responses. But overall, what is the main function of the immune system? And overall, the main function of the immune system is to defend against infectious pathogens. And there are really three main components, or maybe sometimes categorized as two main components of the immune system. In this slide, I've illustrated them as three separate things. So one that's really critical and people don't often associate with immunology is just the importance of our barrier tissues in protecting us against infectious diseases. So barrier tissues include the skin, but also really any epithelial cell in your gut, in your reproductive tract, in your lung that has exposure to the outside world. And we know that if we have if those barrier cells are compromised, for example, in patients that suffer severe burns, we're at a hugely increased risk of developing very serious and life-threatening infections. In addition to these barrier cells that are pre-existing, obviously, and help protect us against infectious disease, there are also various cells and other associated molecules that are also pre-existing or act very, very rapidly that also protect us against infectious disease. And these pre-existing cells and molecules and other cells and molecules that respond very rapidly to infection are broadly categorized as members of the innate immune system. And we'll talk more about that as the lecture progresses. And finally, we have another arm of the immune system is the adaptive immune system. And what we largely mean by the adaptive immune system are lymphocytes, T lymphocytes and B lymphocytes, and molecules that T lymphocytes and B lymphocytes make, like antibodies, for example. And we'll talk a lot more about adaptive immune system function again in later mini lectures. Okay. So just to define, again, the innate immune system and an adaptive immune system, you can think of the innate immune system as a collection of molecules and cells that distinguish host cells from infectious agents, largely by recognizing a relatively limited number of conserved patterns expressed by microbes. These receptors that these cells use are encoded by germline, what are called pattern recognition receptors, and there's not a lot of these receptors. They're relatively limited, and they recognize broad patterns that are commonly expressed on microbes. And I'll talk more about some of these patterns and pattern recognition receptors in a few minutes. The innate immune system largely pre-exists, meaning that it's constitutively present, or that it's activated relatively quickly within hours of exposure to pathogen-associated molecules. And typically, the efficacy and activity of innate immune system functioning is not changed by previous exposure. And so these characteristics of the innate immune system are really different from characteristics that define the adaptive immune system, where the adaptive immune system is defined by immune responses that are mediated by lymphocytes. And in this case, the lymphocytes use receptors that are hugely varied and can recognize a virtually unlimited millions and millions and millions of different antigens or different types of molecules. And these receptor specificities, as you'll learn in later lectures, are not encoded by individual genes, but are encoded by rearrangement of little mini gene segments that occur during lymphocyte development that allow lymphocytes to express a very broad array of receptor specificities. Typically, the adaptive immune system is much slower than the innate immune system, meaning that the efficacy of a response is not usually seen until a few days after exposure, and in some cases it may take a few weeks. And the other thing that's characteristic about the adaptive immune system is that its character, type of response, changes upon subsequent exposure. So the first time the adaptive immune system is activated, you may get a relatively limited, maybe not so efficacious of a response, but upon subsequent exposure, the immune response is much stronger and more effective and more protective against that given pathogen. So just to review in a little bit more detail the role of barriers and molecules that are associated with barriers in protecting us against infectious disease. And so these barrier sites, like the skin, like the GI tract, respiratory tract, reproductive tract, and other tissues that have exposure to the outside world, all have evolved to mechanisms that help limit the invasion of microorganisms into our bodies that potentially could cause damage. And there's a variety of ways that this works. One is strictly mechanical, just the presence of the skin epithelial cells and tight junctions that prevent entry of microorganisms, but also fluid that's produced by these epithelial cells, saliva, mucus, sweat, cilia that may be expressed on the surface of the epithelial cells, all serve to protect us against infection with microorganisms. There are also specific chemicals or proteins, enzymes that are made by these immune cells that also play a role in protecting us against microbial infection. And finally, one of the things that's become more and more widely understood as being critically important in regulating the function of the immune system is that we're all colonized with bacteria at all these barrier sites, on our skin, in our GI tract, in our reproductive tract. And these microbes that exist on all of us are considered normal flora, and they don't normally cause illness. They don't normally cause much of an inflammatory response, but they serve an important function in a variety of ways. So one way is that they occupy a niche that prevents the infection of other microorganisms that may cause infectious disease. But the other thing that they do is that they interact with these epithelial cells, these barrier cells, as well as cells that are just underneath the epithelial cells in the mucosa, immune cells, to help prime the immune system to make it a little bit more active and induce the production of some of these chemicals and immunological mediators that protect us against harmful pathogenic infection. Another group of proteins that pre-exist and can be thought of as being part of the innate immune system is the complement system. What the complement system is really, it describes at least 30 different proteins that are largely produced by the liver and are present, pre-existing at high concentrations in the blood. A large fraction of our total protein component in the blood is made up actually of complement proteins. This series of proteins are very old evolutionarily. Many evolutionarily more primitive organisms have complement proteins, and they seem to have played a role in immune protection in many different microorganisms. There are three main pathways that are associated with complement activity: the classical pathway, the alternative pathway, and the lectin pathway. The classical pathway is actually activated by products of the innate immune system. In order to activate the classical pathway, you need to have antibody interaction with antigen, and I'll define these terms a little bit more in the next lecture. The lectin pathway is dependent on complement components binding to carbohydrate residues on the surface of microbes. And the alternative pathway is again dependent on different complement components interacting with pathogen surfaces, largely because of the charge characteristics, negative charge characteristics of the cell walls associated with microorganisms. Regardless of which complement pathway is activated, though, the end result, or immune function response, is the same. So complement activation results in really three main effector functions. So one would be the recruitment of inflammatory cells to the site of infection. Another would be what's called opsonization of pathogens, so it coats the outside of pathogens and facilitates their detection and clearance by immune cells. And finally, complement plays a role in directly killing pathogens. And I'm going to go through each one of these functions really quickly in the next 3 slides. So this first function, again, of complement is opsonization. And what that refers to, in this case, the little red triangles are coating the little green microbe here. And the presence of the complement proteins, the red triangles, facilitate recognition of the microbe by phagocytic cells, in this case a macrophage, and allows the internalization of this microbe inside the macrophage. And then the macrophage can activate antimicrobial pathways that will result in killing of the microbe. The other important function of complement is that complement proteins, when they're produced, will go out into the circulation and serve a chemoattractant function. And what that means, they have the ability to recruit other innate immune cells, for example, neutrophils and macrophages on this slide, to come into the tissue where the complement pathway has been activated. So in this cartoon, you see a neutrophil, you see a macrophage, and the presence of the complement protein serves to help recruit these neutrophils from the bloodstream into the site of the inflammatory response where the complement proteins are being activated. And finally, complement has a role in directly killing microorganisms. So there are a series of complement proteins that when they're activated, and again, don't pay attention to the numbers here, it's just a series of different complement proteins that assemble together to form a pore on the surface of the microbe, in this case on the surface of a bacteria. And by forming a pore in the cell wall of the microbe, it destroys the osmotic balance of the microbe, and extracellular fluid rushes in and the microbe will burst by osmotic lysis. Okay. And so those are all things that can be thought of as molecules that pre-exist and serve to form initial protection against microbial infection. These molecules interact with cells of the immune system, and as I said before, there are kind of 2 main ways of classifying cells of the immune system. There are innate immune cells and adaptive immune cells. And so there's this long list of innate immune cells, and we'll talk about some of these in more detail in a few minutes. And then there's adaptive immune cells that are primarily categorized as B and T lymphocytes. And this is what some of these cells look like. Some of you have had more experience in looking at some white blood cells under the microscope and peripheral blood smears. And so this is a picture of a neutrophil, and you can see that its nucleus is multilobulated and has fine azurophilic granules in the cytoplasm. This is another innate immune cell termed an eosinophil, and this nucleus also is usually bilobed, so multilobulated, and also has granules that are coarser than the granules that you see in the neutrophil, and you can't really see so well on this slide, but are pinker in color, more eosinophilic. This is a picture of a macrophage. Macrophages, as you'll see in a few minutes, largely reside in tissue and serve to phagocytose other cells and microbes. And so this is a picture of a macrophage, and you can see it's phagocytosing things because it has a lot of vacuolated inclusions in the cytoplasm here. And then here you see lymphocytes. Lymphocytes aren't very interesting to look at morphologically. They're mostly just round cells that are primarily made up of a nucleus with relatively little cytoplasm. And you can't tell the difference between the different types of lymphocytes by looking at the cell under the microscope, but lymphocytes, as you may know, can be divided up into different categories. 2 important categories of lymphocytes would be T lymphocytes and B lymphocytes. And one way that you'll see in a few minutes is we can identify whether or not a cell is a T lymphocyte or a B lymphocyte by the pattern of proteins that are expressed on the cell surface. This is another picture of another lymphocyte. In this case, it looks a little bit different than this one in the sense that this lymphocyte has more cytoplasm and within the cytoplasm, again, has small azurophilic granules, and this is characteristic of a cytotoxic T lymphocyte, or it could also be a natural killer lymphocyte, and those plasmic granules are key to the function of these cells in that they contain molecules that can kill other cells when these lymphocytes interact with those other cells. So as I mentioned, one way that you can tell different subsets of lymphocytes apart is by assaying their patterns of protein expression on the cell surface, or sometimes in the cytoplasm, or sometimes in the nucleus. And a lot of these proteins are classified in what's called a cluster of differentiation system, or a CD system. And so CD stands for cluster of differentiation. And what that means is it's really a basis of a system for classifying surface molecules of immune cells using antibodies that recognize these proteins. And the term cluster reflects the fact that each molecule is usually recognized by a group of previously defined monoclonal antibodies, and the expression pattern of these molecules with different cell types or differentiation states kind of assigns them to different cells or states of differentiation within a cell type. So that's why it's called cluster of differentiation. And so an example of how you use this system to identify different classes or types of lymphocytes is that all T cells, all mature T cells, would express this protein called CD3. And so if you identify a cell that's CD3 positive, by definition that would be a mature T cell. And it's a mature T cell because the CD3 complex of proteins is associated with the T cell antigen receptor. And then T cells can be further subdivided by other patterns of protein expression. So for example, a CD3 positive T cell may express this other protein called CD4. And so you can have some cells that express CD3 and CD4. And the expression of the CD4 protein is associated with T lymphocytes that largely have helper effector functions, but not always helper effector functions. And we'll talk more about that again in a later mini lecture. Another protein that's expressed on T cells is called the CD8 protein. And again, the CD8 protein helps classify T cells with their ability to develop cytotoxic effector function. And so you have these two main classes of T cells, some that express the CD4 protein and some that express the CD8 protein. And the expression pattern of those proteins is associated with the ability of those cells to ultimately develop different immune effector functions. The other example of proteins that are used to categorize lymphocytes that you'll see used clinically a lot of times is that B lymphocytes are largely defined by their expression of this protein called CD19. So if you hear a cell being called a CD19 positive lymphocyte, what that means is that it's a B lymphocyte. By using monoclonal antibodies to identify patterns of protein expression, although it is used clinically relatively commonly, the much more common way of assessing and categorizing different immune cells is by looking at them morphologically in the blood. And so this is just a tube of blood that was drawn from a person, and you can see it has a purple top tube. And what that indicates is that there's a certain coagulant in those tubes, anticoagulant in those tubes that inhibits blood coagulation, so that you can take some of that blood and spread it out on a glass slide and look at the cells that are in that tube of blood. And you can see here there's a mixture of white blood cells that are in that tube of blood. And so based on the slide that you saw before, you should hopefully be able to identify some of these cells. So this is a cell that's an eosinophil. This is a lymphocyte. This is a monocyte that will develop into a macrophage when it goes into the tissue. This is a neutrophil, this multilobulated cell. Here's another lymphocyte. This is a cell that we didn't really talk about called a basophil that has these large basophilic granules. And what you see in this tube of blood is that most of the content of this tube is plasma, so acellular fluid. And then the other part of it is largely made up of red blood cells. So by far the most prevalent cell type in a tube of blood would be red blood cells. And the white blood cells, or the leukocytes, represent a very small portion of the cellular constituents within that tube. But nevertheless, people look at that tube of blood as a window on the cells and potentially functioning of the immune system, and it's a routine laboratory test that's done all the time. And so if you draw a tube of blood like that, you can get what's called a white blood cell differential to categorize the relative numbers of neutrophils, eosinophils, basophils, monocytes, and lymphocytes that are in blood, and this just represents kind of the normal reference range of these various cell constituents. And what you can see is that neutrophils represent typically the majority of white blood cells that are in blood, followed by lymphocytes, followed by monocytes, and eosinophils. But the thing that I want to emphasize here is that these cells, depending on the cell type, may represent a tiny fraction of the total white blood cells that are in the body. And this is especially true for lymphocytes. There's only a tiny fraction of lymphocytes actually reside in the blood. Most lymphocytes reside in lymphoid tissue. And so this is just a slide to get across that idea in a little bit more detail about where do cells of the immune system live. And it's a little bit complicated, and it depends in part on the state of development or state of activation of a given cell. So all of these white blood cells arise from a common hematopoietic progenitor cell that's in the bone marrow, and all of these different cell lineages develop at one stage or another within the bone marrow. And so you have a common lymphoid progenitor that's in the bone marrow, and some of these cells will commit themselves to developing into B cells, and those cells will further develop into mature B cells in the bone marrow. But some of these common lymphoid progenitors will leave the bone marrow and move to another lymphoid organ called the thymus and develop into T cells. And then yet another lymphocyte subset that we haven't talked about yet is a cell called a natural killer cell that also arises from that common lymphoid progenitor cell. And pretty much all of these lymphoid subsets, although they will recirculate in the blood, they predominantly live in lymphoid tissue like lymph nodes or spleen or tissue that we'll refer to as mucosal associated lymphoid tissue that I'll talk about in the next mini lecture series. Other white blood cells that also arise from this common hematopoietic stem cell precursor will develop in the bone marrow and develop into granulocytic lineages of cells. So for example, neutrophils, eosinophils, or basophils. As you'll see later on in this lecture, neutrophils that develop in the bone marrow, they predominantly leave the bone marrow and recirculate in the blood and will only go to tissues when they're called to tissues in response to an inflammatory insult. Whereas other cells, like macrophages, mast cells, cells called dendritic cells, actually don't tend to reside in the blood to any significant extent, but constitutively reside in peripheral tissue where they're exposed to microbes that get through those barrier tissue sites. One other thing I just wanted to mention is that although lymphocytes predominantly reside in lymphoid tissue, when lymphocytes are activated in this lymphoid tissue, as you'll learn in later lectures, they will proliferate, make more of themselves, and differentiate into cells that we refer to as effector cells. And often these cells will leave the lymphoid tissue and migrate into peripheral tissue sites at the site of active infection.